Hello, everyone, and welcome to today's webinar. My name is Dr. David Taylor. I'm just going to get verification from outside my office that people can hear me. And I don't have any verification, but I'm going to keep going and they will tell me if you can. So welcome to today's webinar. Today we're doing this in two parts. In the past, I've done it in one part, but I feel like we've had to rush through some critical issues in the past and some critical messaging. So I really wanted to separate this into two pieces, and I'll explain what those two pieces are as we move forward. Before I just kind of get this presentation working. There we go. Before I get into any of the actual information, I have two things here, which are slides which reference all of the images that you'll see. This is purely because we do want to be able to make these streaming and on demand for people to be able to go back through them, or if you're unable to attend today. I want to make sure people get proper credit for the images that are used on that streaming option. Now, this is something I've never done before on these webinars, and I wanted to start off by just very briefly explaining who I am. And the reason why is because there are many sources of information out there for people living with ALS, and I really wanted you to be able to get a feel for who is telling you this information. And the basics here is that, I worked on ALS as a laboratory researcher for 12 years. And then have now had the fortune of working at ALS Canada to run the national research program for the past 7 years. And the key piece I really wanted to relay is that, a lot of the answers that you'll get here, a lot of the information that you'll get here is probably not what people really want to hear. And that's frankly quite awful. Like, I wish we had better answers in a lot of cases. So I wanted to be able to provide an opportunity for compassionate, direct answers to your questions. And I wanted to let you know that, if there was something real out there that worked today, and if we knew it worked, we would tell you. I would be telling you on this webinar, and we would absolutely be trying to get that to people as quickly as possible. And the other piece is that, I don't know everything about ALS. I know a lot. And I think you can always validate the things that I say with other sources, and I'm always happy to discuss things. And certainly at the end of this webinar, I'll stick around as long as it takes to answer your questions, and you can follow up with questions afterward. Very briefly, what is ALS? The reason why I have this is because we don't know. Maybe some people who are attending the webinar, it's their first time coming to any kind of ALS webinar. So just like to explain that ALS is essentially a disease where the living wires that are called motor neurons that connect our brain to our muscles degenerate and die. So each muscle cell in our body is connected by one upper motor neuron and one lower motor neuron, which allows for that muscle to contract. And as these degenerate and the connection is lost, you can imagine that that causes that muscle cell to paralyze. As this paralysis spreads throughout the body, eventually it would reach the muscles that allow us to breathe, and this is what causes it to be a terminal disease, often about 2 to 5 years after diagnosis, but this can be very different from person to person. And we'll certainly talk about that moving forward as well, because that has a big implication on clinical trials. It's a very crude diagram to show you. In black, you have your upper motor neurons, and they extend down from the brain into the spinal cord, and then a lower motor neuron will leave the spinal cord and go to each of the muscle cells of the body. And bundles of these motor neurons will have electrochemical signals that will move along that pathway and tell those muscles to contract. And then in ALS, as you can imagine, when these living wires of these motor neurons degenerate, those connections are lost, and that's what prevents the brain from being able to tell the muscles to move. And one other thing we're not going to really focus on in this webinar, but is very important to know, is that there's not just dysfunction happening within the motor neurons. There's actually a lot that is known around of the cells that surround motor neurons and provide the environment in a normal system, a nurturing environment for motor neurons to survive. And these cells are also a part of the pathology of the disease and can be targets for clinical trials and for experimental therapies as well. Now, this is something that I typically will do in a full webinar, but I'm going to shorten into one slide here, and that's really a state of where ALS research is at in terms of at least preclinical research in the laboratory. And I've really condensed it here. If you start in 1939, this was where ALS sort of got wider prominence beyond being known by just neurologists, as Lou Gehrig was diagnosed and passed away 2 years later. We really wanted to start ALS research after that. The problem was that technology didn't exist to do it really well. So the only things that could be done were observation of people living with the disease and what we could learn from tissue that was donated generously after people passed away. But it wasn't until 1993 that we had sort of the big breakthrough, which was the discovery of the first genetic cause of ALS. For some people with the disease, they will have a genetic mutation in something called SOD1. And what was really great about that is it gave us a tool or a puzzle piece to work with in the laboratory that could allow us to make models of the disease and start to study this in the lab. But it wasn't until 2006 that we really sort of hit the genetic era of ALS, and we started to discover more of these genetic causes of the disease. And then by 2012, we discovered a very big puzzle piece. And in the subsequent 7 years or so, we've discovered most of the key tools that we can use now to try to understand and plug them together to make models of the disease in the lab and ultimately try to understand why is ALS happening, causing our motor neurons to degenerate, and that's ultimately going to correlate into better clinical therapies. This is just to show you again the advancement that's happened over the past 5 to 7 years or so in terms of those tools that we can work with to understand the disease better in the lab. You can see that in the last 5 or 7 years, we've discovered so many of these tools, and this comes back to the fact that ALS is a grossly underfunded disease. And a lot of these haven't been explored in heavy detail, largely because we don't have the resources to be able to work on them in the same way as other treatable diseases may have. Also, this is just a diagram to show you that we have actually come a very long way in our understanding of ALS, and there's still a lot to understand. It is a very complex disease, and ultimately, if we look back even just 5 years ago, we probably would have had a much smaller diagram from where we are today, and each one of these different targets are things that we might be able to create drugs or therapies that can actually slow progression of ALS once we're able to circumvent what's causing the motor neurons to degenerate. So this one's titled Doing Our Best, and part of the reason for this is that often people wonder, well, does anyone fight for ALS? And absolutely, people are fighting very hard for ALS. And one of the things about the field of this disease is that people are doing their best with very little comparatively to other more treatable diseases. So this is an example of an annual report from the American Cancer Society. You can see here that an annual level of funding is about $145 million just from one wing of cancer research. Here's the Towards an HIV Cure Fund from the International AIDS Society, showing that in 2016, $268 million could go into HIV cure research. And now if you're looking at, say, the ALS Association, because we're comparing US societies here, this is obviously the biggest group for funding research in any field. And you can see that before the Ice Bucket Challenge, the annual levels of funding that can come from ALS in the ALS Association, it's around $3 to $6 million. So that doesn't go as far and doesn't allow us to do quite as much as you could with a large amount such as the other ones. Now, certainly, that's not to say that we don't need those levels of funding in those other ones, but ALS not only has less, but it's certainly a more complex disease in that we don't even understand the central nervous system fully and are trying to understand how this dysfunction occurs. Furthermore, the Ice Bucket Challenge was a tremendous gift, but it needs to be taken very carefully, as this is a one-time gift that still pales in comparison to the annual amounts for more treatable diseases. So we are doing a lot with a little, but that always needs to be taken into context when it's considered, how well we have to be very careful with our funds. So as far as part 1 and part 2 of this webinar, I'm actually going to take the opportunity here in part 1 to talk to you a little bit about various questions that I've gotten over the years, things that I've kind of whipped through in various webinars in the past where it's been 1 hour, and I'm going to take a little bit more time for that to build a foundation so that next week in part 2, when we talk about specific experimental therapeutics that are happening in ALS, we'll be able to get a better focus on, you know, some of the nuances and understandings of where those really are in our understanding of whether they work or not. And also next week, I would like to talk a little bit about some of the tools that can help you to be involved in clinical trials, and also to be able to navigate certain things that are happening, whether or not they're legitimate trials or not. I'm just going to mute myself for 1 second because I'm just going to close a door so that it's a little quieter. Okay, sorry, we're back, and now we'll get this going again. And okay, sorry, everyone. Okay. So another foundation I wanted to provide, which helps us to interpret where clinical trials are at, is thinking about the motivation to succeed from each of the different groups or stakeholders who are involved in clinical trials. And so the first one I'm going to speak about are companies in pharma. And we have to remember one of the key aspects in interpreting clinical trials is that they are the owners of most of the treatments. So when it comes to an experimental therapeutic that may be showing promise in ALS, we have to remember that they are owned by companies who have investors to try to ultimately get a market and create return on investment for those particular therapies. Now, this is by no means any way to demonize these. They are our partners in being able to get therapeutics to people with ALS. Almost everyone I've ever met who works for companies and private companies and pharmaceutical companies truly do care about helping people living with the disease. But we also have to remember that press releases, which you may read online, are not peer-reviewed publications, meaning someone can say anything they want. And we have to remember that in the end, these companies are beholden to investors who are looking at the bottom line of a business, who are trying to ultimately get that return on investment. And this is a very crass example, but it's again to sort of show you that there's no difference other than the fact that these are viable therapies that biologically we hope will work. But there's no difference from somebody saying, listen, I have a degree and I want to sell you this magic lemonade, and this magic lemonade works for ALS, and I know that it works, and I want to sell it to you for a whole lot of money. Not saying that these are the things that the companies are doing, but I'm saying that in a situation like this, we do not know necessarily whether something works until it is vetted scientifically. Now, the second one is governments or Health Canada. Now, we have to remember again that if a company owns a particular treatment, they can only respond to applications that come from those particular companies or from pharmaceuticals that own the actual treatment. And in their case, they want to keep Canadians safe and not taken advantage of. And now I understand that, you know, a person living with ALS may not, the safety is not necessarily the biggest concern. And especially if something is considered safe, they want to be able to try it. And we'll talk about that as I move forward. But the other piece is that there are a number of dubious actors out there in ALS, not the companies I was talking about before, but there are a lot of clinics and other physicians out there around the world who will sell therapies that are not proven, but they will try to tell you that they are proven. And so Health Canada does want to try to govern some of that stuff, because not everyone's going to be able to know what the difference is between something real and that something's not. Everybody that I've met in any kind of government position really doesn't want to put unnecessary barriers up, and they're really trying to walk this fine balance between, you know, taking care of people from their standpoint, and also not putting up these unnecessary barriers. But there are definitely things that can be done that can better suit ALS, and we need to continue to advocate and educate our government and Health Canada to find better ways that can suit ALS specifically, because nothing will ever be fast enough for this particular disease. And in terms of clinicians, researchers, ALS nonprofits, really, they just want to make sure, again, that people are not taken advantage of by some of these dubious actors, but also in the sense, these are really hardworking, compassionate people who choose to fight for ALS. And, you know, there are other avenues in which people can go to, but they really want to work on ALS because they want to be there and figure something out for people who have the disease. And I can say without a shadow of a doubt, I have met some of the hardest working people who are focused on really trying to fix ALS. If they knew, again, of a way to be able to find something faster or better, they absolutely would tell people. So sometimes when people say the truth is out there, that may not necessarily be the truth itself. And so we always need to be careful about what's read and try to think about all the different avenues and still try to think about how we can get things to people as quickly as possible. Everyone on all of these really do want to get something that works to people as soon as possible. So in every case, it's either purely from compassionate means, and in some cases, it's also because the faster it gets to people, the quicker they can, a company can start to make return on investment and have a market for all the money that they put into the clinical development of a particular therapy. So nobody wants this to be held back or slowed to actually getting to people as quickly as possible. So getting into some of the details, what is a clinical trial? Very basically, that's just any experimental study that happens in humans living with ALS. They could be things where we're learning about ALS, so observational studies where we're learning more about the disease. Very important studies are done on better ways to manage symptoms. So for example, perhaps a therapy that can reduce salivation or something that can prevent the twitching and the fasciculations. These can all be really important things to study in clinical trial. We're going to focus very heavily in this particular case on interventional clinical trials, ones that are aiming at slowing or stopping the disease progression. So we also have to remember, and I will reiterate some of these messages many times, is that a company or an academic grant pays to do a scientific experiment. That's ultimately what a clinical trial is. And so, first of all, if you are involved in a valid clinical trial, you are not paying to do anything. That is something that is covered by the cost of the clinical trial for the person who is running the actual study. The questions are, is it safe? That's the first big question. And is it tolerable? And then the second piece is, does it work? And the key thing is that if it is in clinical trial, then it is something that nobody knows the answer yet as to whether it works. We don't know, nobody does. So if anyone tells you that they know it works, then that can't be true, or else they would have applied to have this reviewed so that it could actually become a therapy approved by Health Canada or the FDA. And this is one of those messages I told you about, and I know it's not a great message for a lot of people to hear, but ultimately clinical trials are a way of contributing to science. They are a philanthropic endeavor, and I can imagine, if it were me, you absolutely want to be part of the clinical trial that is going to be the first one that's going to have a significant effect on ALS, and also be a part of the non-placebo group. And we'll talk about placebos as well today in the webinar, and why they still have to be important. But ultimately, it is a way of contributing to science so that we can ultimately find therapies that truly work for the disease. So just going through very quickly which types of trials exist. I'm sure many people know this already. Phase 1 clinical trials are really about safety and tolerability of a particular therapeutic. Often these are multiple doses or regimens to try to figure out what is the best and safest option to be able to move forward to a larger trial. It's really designed to just pick a dose to be able to go on to better trials for a company. So you have to imagine that each of these clinical trials that they would run are going to be progressively more expensive because there will be more participants within them. So to be more safe within an investment, they typically will do a smaller trial first, make sure that they've got the right dose, because when they move to the next more expensive trial, they want to make sure that they picked it effectively in the phase 1, as well as obviously needing to be able to show safety and tolerability. Moving to a phase 2 clinical trial, this is really still around saying, now we've got our chosen dose, we're going to do more people, we're going to make sure that it's safe and tolerable, and then you're going to hopefully get that first little indication as to whether it's something that they want to elevate to a phase 3 or a larger clinical trial. However, in most of these cases, they're what we call underpowered. So the phase 2 clinical trial is something that often is not statistically significant enough to really derive a good answer. And I'm going to talk about this actually quite at length in today's webinar about some of the times where we've seen phase 2 trials, and we have to be very careful about how we interpret these underpowered trials. You can also see there that this already often gets into many millions of dollars to run an effective phase 2 clinical trial. And you can see that for nonprofits like ALS Canada or ALS Association, it's very difficult with limited funding levels to be able to fund these clinical trials without doing a complete removal of the amount of funds that go into helping us advance our understanding of this very complex disease. So this again shows you why it's a partnership with pharma and with biotechs to be able to say they need to be the ones that push this across the finish line in a lot of cases, but we need to make sure that we work together to do that as quickly and as effectively as possible. And then phase 3 clinical trials, this is really that we're going for it phase. This is what we often call a registration trial. This is where they're statistically powering it up enough so that they can get an answer as to whether it's effective at slowing down the progression of ALS. And as you can see, these are quite expensive trials. And so often when you're thinking about it from a business standpoint, and that's who's running most of these trials, they need to make sure that all their ducks are in a row before they get to this phase 3 trial so that they can make sure they get a definitive answer as to whether it works or not. So if it does work, they'll have the right answers to be able to apply to FDA and Health Canada to ultimately get the market that they want to have. And we, as individuals who just want therapies to happen and to come to ALS as quickly as possible, want this process also to happen as effectively and as quick as possible. But all of that to say also, there is really hope for the near future. In the next year or two, we're going to be able to get answers from a number of phase 3 clinical trials. Never before have we had this many phase 3 clinical trials at one time in ALS, and I'm going to talk about each one of these individually next week in the webinar on the specific experimental therapeutics. We're also starting to see a number of unique experimental treatments in the early stages of trials that we've never really had before. So most of the substances that are in phase 2, late phase 2 or phase 3 clinical trials are things that are based on a more high-level understanding of what ALS is, but they're not necessarily nuanced on our greater understanding of the molecular disease that has occurred over the past several years. What we're starting to see is some of those phase 1 or phase 2 trials coming in that are actually related to some of those really interesting targets that have developed out of our better understanding. And I'm going to talk about some of those as well next week. The other big piece, and I'm going to really talk about this a lot as well, is that, we've learned a lot in ALS from our past failures, and we are not duplicating those. In fact, we're trying our very best to put effort into making trials more efficient and more effective based on what we've learned over the past. So this is just to give you an idea of some of the things that are in the clinical trial pipeline. I'll be talking about a number of these next week in more detail. But again, to show you there are a lot of things that are already in the phase of human testing, and any one of these could potentially be something that will ultimately prove to slow down ALS. But you can also understand that when there's this many things in trial, and we have had a fairly high failure rate in ALS, a very high failure rate, it does explain why we need to be very careful about interpreting anything before it's gotten to a very definitive experiment in a phase 3 setting. So as I said, one of the things I'm going to do is not necessarily talk about the things that I think you need to know about, but also try to talk about some of the things that I've been asked many times over the years. And again, why I would like to prompt anyone out there to say, if you have other questions that don't get answered today, please feel free to address them directly with me after the webinar. One of the questions I frequently have is, why do other countries have certain clinical trials and certain treatments that we don't have in our given country? And here in Canada, that's the particular question that I would get. You have to remember again that companies own treatments. They decide where to run trials and to apply for approval. And Canadian clinicians are always trying to push and get promising clinical trials here at Canadian sites. The CALS network, which is the Canadian ALS Research Network of clinics, are continually trying to bring the best and most promising clinical trials here to Canada, including stem cell trials. And I'm actually going to use an example here of to try to explain this situation in a more pragmatic way. So I'm going to use the example of Brainstorm, their treatment Neurone, which is a stem cell treatment in phase 3 clinical trial. A lot of people ask, why do we not have this here in Canada? Well, they ran a phase 2 clinical trial at 3 sites in the United States, 2 in Boston and one site in Minnesota at the Mayo Clinic. And you can imagine that when they wanted to run a phase 3 clinical trial, they have limited dollars to be able to run this trial effectively. First of all, the infrastructure was already set up at 3 different sites in the United States. They wanted to expand to 2 more sites in California, but they've also already had a relationship with the FDA. And so pragmatically, from a business standpoint, they chose to do 5 sites in the United States. And to their credit, they actually pushed to try to get Canadians eligible to be able to be involved in the clinical trial. So in a sense, that is why we don't have a clinical trial like that in Canada, because the company ultimately will choose what is the best way that they can most efficiently and effectively, in their eyes, get the ultimate answer that they need to get. Just as an example, when people often ask, well, why do we have to travel to the United States to get something that we feel might be promising or might have potential? Well, actually, I was talking with a society from Argentina recently and realized, in a lot of countries like that, there are no clinical trials that they have access to. And also, when you think about big major cities in the United States, like Houston or Chicago, for example, they also would have to travel to be a part of a clinical trial like that. So there's by no means Canada being left out, and other countries in the same way. It's really a way in which a company tries to decide what is the best way that they can run their trial as effectively and efficiently as possible to get the answer they need to get. And in terms of actual treatments, I can definitively say that there are no proven treatments for ALS available in other countries. Now, the exception of that is Radicava or Radicut, which has been proven in the sense that it's approved by Health Canada, FDA, South Korea, and Japan, but it is still not actually available here, but it is approved by Health Canada. However, when we look at the other things that are approved in other countries, for example, Nuedexta, which is a symptom management drug for what's called pseudobulbar affect, the laughing and crying that can be associated with some people's ALS. This is not something that the official drug needs to be necessary, that clinicians can manage this at some level here without needing the official drug. And also, another question that I often get is around something called NeuroNATA-R or CoreStem, and I will be discussing that again next week. And that is not something that has definitively been considered proven yet. And there are definitely nuances that I'll talk about next week about the results that we know to date that show that we do not know officially whether this works. And also, in this sense, that company, again, the owners of the treatment have not applied to North America and do not have a clinical trial happening in North America. So again, it's a situation where it's not necessarily that Canada or our government or our Health Canada is not doing something. It's ultimately that the owners of treatments decide where they want to be able to take that treatment to go forward. Another question that I frequently get, and I'm going to focus on this a little bit more today, is why does it take so long for clinical trials to happen? Now, we have to remember that clinical trials are actually science. And in fact, one of the disservices that has been done to science is the low-hanging fruit that has existed for years. So penicillin and the wow, eureka moments are things that happened in the past, but we're farther away from those now. Getting those kind of magic bullets in the central nervous system, which is much more complex, is something that we very likely won't have. It's possible, but people expect those, but science takes a lot longer and is more rigorous. Things like CSI, where someone just does a couple experiments and suddenly they know who the murderer is. That's just not how science really works. And the word breakthrough, these have all done a little bit of a disservice to the wider population of understanding how rigorous and difficult it is to do science. There are really a lot of logical steps that have to happen to prove a treatment to work. And I'm going to go into those in a bit of detail right now. But I have to tell you that people working on ALS are doing everything we can do to try to find as many efficiencies as possible to speed this up and still get definitive answers as to whether something works. So I'm going to go through this in a way I've never done it before. Hopefully, I'll keep people with me. It may be hopefully not too boring, but the idea is here, really, what does it take to get from an idea through to a prescription? Often someone will just say, well, it takes 10 years or it takes 20 years, but maybe haven't delineated what that actually involves. So if we start just from the first section of trying to take something from an idea through to a clinical trial, well, the first step is, let's say someone has a great idea in the lab to work on ALS. They have to write a grant. They have to go through a competition to make sure those valuable funds are given to the right study and receive the funding. Starting a study then, let's say a very quick study in zebrafish, which can get paralysis when you give them a genetic form of ALS. And let's say that the person hypothesizes that some fake factor Z is harmful to motor neurons, and if we inhibit that factor Z, it might actually slow down progression of the disease. And let's say after 2 years, it works in the zebrafish and they're pretty excited. Well, in order to be able to move that forward to humans, typically Health Canada, FDA, they're going to need a little bit more proof than that. So let's say the scientist then collaborates with another scientist. They meet at a conference and they say, let's do this in your mouse model of ALS. They want to be able to test certain drugs that inhibit factor Z and see if that'll work in the mice as well. So again, they have to take time to write a grant. They have to get a competition and funding. And then after 4 years of testing these various compounds in the mice, they ended up being able to publish this. And let's say that a company called some generic biotech has something called compound S that actually blocks and does the same thing as this particular therapeutic that they saw in the lab. So they decide to invest in this particular idea and try to move this forward to clinical trials. Well, they need to do fundraising, so they need to be able to ultimately get the money together to be able to run the clinical trial. And then there are a number of steps that actually have to happen that people don't often talk about to be able to take something from, say, a lab powder all the way through to something that can be given to humans in a clinical trial. So this involves such things as making sure it gets to the target through the what's called the blood-brain barrier. How long does it stay in the body? Is it toxic to people? Is this something that we can prove without a shadow of a doubt to Health Canada? It's going to be okay to put this in humans without causing significant harm. And so right now you're talking about 12 years just to get something from idea in a very ideal situation through to a potential clinical trial. Now, if we move forward in clinical trial and we want to take that through to the Health Canada timeline where you've got something that works and is proven in clinical trial, let's say we've got our company now and we've got something we're going to call ALS Z because it's a factor Z inhibitor. Now, factor Z is just fake, but it's just to give you an example. We want to put that into a phase 1 clinical trial, and we're really going to be testing 5 different doses at 5 sites and be able to say, is this toxic and tolerable for people who are living with ALS? From the time that you start and design the trial all the way through to reporting in a very, very ideal situation, let's say that's another year. Now, something we're going to talk about more detail that you may have heard of, copper ATSM, this is an example of being just done this particular type of study at this point. I'm actually going to jump away from that timeline and I'm going to go on to something that I know a lot of people ask about, and it comes from this idea of being at the end of a phase 1 clinical trial and wanting to have a right to try. If we know it's safe, why can people not try it with ALS? And I think one thing I can say is that most people who work on ALS at any level of the different groups that I've talked about, if they had the similar situation, they would also completely understand the desire and want to be able to try something if you know it's safe. But I'm going to try to just explain to you some of the nuances as to why each of the stakeholders have why it's a little more complicated when it seems like a very simple idea from the outset. So in terms of governments and Health Canada, I can say for the most part, they really do want to find ways to help people get access to these therapies for diseases like ALS. And if you look, for example, in the United States and a lot of places where right to try has been passed, there actually hasn't been a lot of uptake in the end. There hasn't been a lot of people accessing therapies. And on the next slide, where I talk about the pharmaceutical company and the company group, you can see why some of that is possible. What are the concerns from a government and Health Canada standpoint? Well, dropping the barriers opens the doors to dubious companies. There are actually a lot of clinics and individuals out there who will say, we've got a therapy that we know works for ALS when they don't. And in the end, this then kind of gives them free rein to sell therapies to people living with ALS. I'm not talking about the ones that are in valid clinical trials right now. I'm talking about ones outside of that I'll refer to more in next week's webinar. Also, allowing companies to sell treatment to the public, it means allowing people who live in that country to save, fundraise, and spend on medicine that very likely will not work. And why do I say very likely will not work? It's because in most cases, we've seen across the board that a lot of therapies fail in ALS. And even though we've seen promising data in early stages, often they don't work. And so having people fundraise their life savings and their family and friends' life savings to go towards something that doesn't work in the end and maybe overpriced is something that certainly Health Canada and the government can worry about. Now, thinking about clinicians, so your physician or ALS nonprofits, again, they're extremely compassionate as to why someone would want right to try. And they do have some of the similar concerns as the government, but there's also the fact that if there was widespread right to try, this actually could prevent us from ever getting to a vision of a future without ALS. Because if all of the funds were going to therapies that work just a little bit or don't work at all, which in a lot of cases may be what happens, there will be no funding in this limited funding realm to actually help us to understand the disease well enough to get to those better therapies and to that cure someday. So it's very realistic to say that if we had a way in which companies could sell therapies to people without them having any proof that it actually works, significant scientific proof, we may actually never get to that future without ALS because we'll have no funding left over anywhere in the field to be able to help that very difficult understanding of this complex disease. But that doesn't take us off the hook from advocating as hard as possible for something that is clearly working and to getting it to people as soon as possible. From the other side of, I just want to give you a couple examples of situations where people have asked for right to try in the past. In 2008, there was an Italian study that showed that lithium delayed progression of ALS in a smaller study. And people really wanted to get lithium, and a confirmatory larger study that happened in Canada and the US actually showed ultimately that it was not effective for the disease. The same thing with a very prominent trial by Biogenetic in 2012, a phase 3 clinical trial based on phase 2 results that did show promise at slowing disease by about 30%, ultimately did fail as well in ALS. And even more recently, a drug called MP001, you can see I've highlighted here that an analysis after the trial showed that about 27% of participants halted with their disease progression. And a follow-up trial that was actually looking specifically at those people that were responders ultimately didn't work as well. So we have to be very careful when interpreting results that are not fully statistically significant yet. And even in terms of other less rigorous clinical trials, we have examples where some companies have done very small trials and given media releases to say how well it works, and people have asked for very quick FDA access to these or Health Canada access to these drugs for a lot of money that they would be sold for, and ultimately have no proof that they work. And I'm not going to talk about this one directly, but I urge you to maybe go read about GM604 as an example of something that we still don't have any evidence works, but a number of people were asking in the past based on very limited evidence, and no evidence, but very limited press releases that this was an effective therapy for ALS. So when we get to what are the concerns of companies and pharma, again, basically we have to remember that companies own the treatments for the most part, and they're beholden to their investors to make a profit at some point. And we also have to remember that legitimate companies are truly compassionate about people with ALS, and they don't, I'll skip to that last concern there, that many of the companies have their reputations based on quality of their products, and they really do want to put something out there that works, and they don't want to tell people about something that they don't know works yet. And so if they were to offer right to try and it didn't work, because they don't know at this point either, it can often infect their entire business well beyond other indications besides ALS. Also, there's a concern that if they provide a drug outside of a controlled clinical trial, this can open up the risk of somebody derailing their chance to go to market. So if there was an adverse event of somebody on a right to try, for example, then this might actually prevent the therapy from ever getting out there. And if it was something that worked, then that actually could prevent it from getting to anyone in the future, or at least significantly slow down the process. And when it comes to the other piece, it comes down to often what is the cost of the particular drug. And a lot of smaller companies can't afford to actually provide right to try to a wide group of people. So this means they will need to sell it at either a break even or a profit. And again, this can be a situation where you can end up drying up the potential for any kind of therapies to move forward in the future if it turns out that this doesn't particularly work. So again, I say these things and I know they're probably not what people want to hear, but I'm trying to compassionately tell you that I think everyone would love to find a way to make this happen, and also considering some of these nuances, how we could make a better system for all someday in the future. But there's certainly significant issues that arise in terms of having this actually work effectively. Getting back to this, I'm going to move quickly because I know I'm down to about 20 minutes. You know, if you finish a phase 1 clinical trial, often an organization needs to then go fundraise and design the setup of a phase 2 clinical trial. This is where often they can talk about the positive aspects of a phase 1 trial, even though there's no scientific or statistical significance yet. You have to remember that they have to find investors for a bigger trial, and often these are small companies that do not have that kind of money in-house. Moving to a phase 2 clinical trial, you know, these are often need to be a bit longer because you need to be able to see progression of the disease. We'll get into eligibility criteria in a moment or two as well. Let's say it's a 200 participant trial. You try to reduce the number of placebos. Again, I'll talk about why placebos are important moving forward in a couple slides from now as well. And let's say at a best case scenario, this takes 2 years to recruit. You have to run the study for the 6 months and then report the results. And then let's say, yes, it looks promising enough to move to a phase 3, but the company's not big enough to be able to afford, say, a $50 million clinical trial. So over the course of the next year, they have to try to set up a deal with, say, a big pharmaceutical company that wants to buy the therapy and then move this forward in a phase 3 clinical trial. And now they're really going to go for it and say, we've got the doses we want. We want to be able to test our best chance at seeing whether or not this actually works for ALS. Doing a large clinical trial at multiple sites, trying to recruit this as quickly as possible, but certainly making sure that it's designed effectively to get an answer. And let's say then after those 3 years, they then are able to show ALS, our fake drug, has a clear progression slowing of 33%. And now it's ready to be applied to any one of the governing bodies like FDA or Health Canada for approval anywhere in the world. And so that's another 8 years. And now we're already up from that idea to the time that it could get to Health Canada in an ideal sense of 20 years total. Now, one of the questions people will say right after I've talked that is, why does it take so long between and during clinical trials? Well, let's take an example where you've got a successful phase 2 clinical trial. You want to move it through to phase 3. Well, a company may have to fundraise to be able to pay for that phase 3 clinical trial. The design is super important, and the reason why is it really has to be perfect to get an answer. And I won't have as much time as I would love to. I can almost do a full webinar on the heterogeneity of ALS, but I think everyone out there can realize that ALS is such a heterogeneous disease in that some people have cognitive impairment while others don't. Some people are fast progressors while others are slower. Some people start with speech and swallowing issues, and others will start with weakness in one of the limbs. And so you really have to be very careful about how to design the trial to make sure that your answer is something clear in the end, so that the company or the group can apply this to be proving that it actually works for the disease. You have to find the sites that you want to run your clinical trial at. You then need to get approval from each country where there are sites, because if you're in Europe, you need the European Medicines Agency to approve your trial, FDA in the United States, Health Canada. You have to go through and make sure that it's approved to actually be done in that country. Then you have to negotiate with each site. What are the costs to be able to run the trial at each one of those sites? Certainly from a for-profit point of view, they're going to need to negotiate to make sure they can get the best possible cost so that they can be able to run the trial as efficiently, as effectively as possible. And clinics will also have to push back to make sure that they're getting enough to be able to cover the cost of actually doing the trial at each of the sites. Then, if you would imagine, say, a trial that works here in Toronto at Sunnybrook Research Institute or in Sunnybrook Hospital, they would need to get approval from the University of Toronto to make sure that the institution in Sunnybrook, that the institution is okay with the ethics of that particular study being run at that institution. These are all things that take time in between the trials to make sure they're up and running effectively. Then there needs to be the time to recruit the people who meet the right criteria, find them, get them into the trial, monitor progression over time, and analyze the data. I am certain that a lot of this is very frustrating to hear. And again, if there was a faster way to be able to do this, we would do it. And I think people are working very hard to try to make these more efficiently and to cut some of these timelines as much as possible. Another question that comes up at this point, before I move to the next slide on timelines, is why are there not more ALS trials with interim analysis? Why do we not stop trials halfway and say, let's just look at the data and see if it's working? Prominent examples that have led to a lot of these questions are that in a disease called spinal muscular atrophy, you've seen two drugs recently, Spinraza and Zolgensma, that have both been stopped early because of the great effects that have been seen. And I actually skipped down here to show you that the reason why, in part, that you could do that with those particular trials is that they had undeniable effects. You were seeing a terminal disease in babies that ultimately changed them from being terminal to not just living, but thriving and meeting the same milestones as other children. So in that case, you know, the risk of a company stopping their trial and looking at it in term is something that they were willing to take because it was so clear that it was working. However, in most cases, these actually need to be in the trial protocol and approved prior to even starting the clinical trial. And you can imagine that if you unblind anything, or if a company starts to look and understand what's happening halfway through the trial, this can ultimately bias what's happening with the rest of the trial and might ultimately ruin the whole thing in the end if it's not very carefully done. So there are real issues and troubles with being able to do these interim analysis. Unplanned interim analysis, again, are big risks to companies, and again, they are beholden to investors, and they want to make sure that the clinical trial at the end gets a definitive answer. And with ALS, it's specifically something that we have to consider as being very different, in that it's not just looking at, say, a tumor shrinking or remission of a disease. You're talking often about moderately slowing a disease that is very heterogeneous. And this can be very hard to prove even through the entire length of a trial with all the participants and all of the time length that was involved in it. And we also don't have large numbers of participants in a lot of these trials. So it can be a very big risk to be able to stop a clinical trial and actually be able to see anything halfway through. But certainly can talk about that in more detail later if anyone has more questions. Moving to that example timeline, I'm gonna speed up here in part 3, basically just to say that if we move through to prescription, there are a lot of things that need to happen to be able to make that happen here in Canada. First of all, most companies will apply to the FDA first. This is the major market that exists, or Europe as well, Health Canada being a smaller market, or Canada being a smaller market. So let's say 6 months after an approval for FDA, and then typically a company might apply to Health Canada somewhere down the road. Now, we will advocate at ALS Canada to say, can we get parallel applications to Health Canada for any promising drug that is proven? But again, you know, it's not the first thought on a lot of individuals' minds or a lot of companies' minds. Even going through priority review, this is a 6-month process after application. And then there are a number of steps that have to happen before a company would likely even launch a drug in the country, and including a review by several bodies called CADIT or INES, which will help to inform whether provinces should potentially cover this on public payer systems and the provincial healthcare systems. And also something called the PMPRB, which basically sets the price at which a company is allowed to sell their drug in Canada or their therapy. Now, if the PMPRB sets a price that is viable for the company to make a profit, they may actually launch the drug and start selling it through private insurance or directly to people who have a lot of money. But if they do not have a price that is set high enough, quite often this can prevent them from actually launching the drug within Canada. And then even once that happens, in order to be able to get it covered on a provincial drug or health system, This often takes many years for each of the provinces to individually decide whether or not they want to put it on what's called the formulary. And so now we're talking many years after the time that something's proven in phase 3 that it actually can get into the hands of someone through a provincial healthcare plan. Now, this is something where we can advocate very strongly at ALS Canada to shrink this timeline and to make sure that government and the bodies that exist can know that this is not effective and not appropriate for ALS, because we certainly need to make sure this gets to people as quickly as possible once there's something proven. And I can say that there is some headway being done, but there needs to be a whole lot more because we certainly need to get things to people once they're proven as quickly as possible. In terms of how clinical trials are run, I'm down to the last 10 minutes. I'm just going to reiterate a few things. I'm going to talk about some stuff that's probably less important. It's really how we determine whether or not a clinical trial works. Less important, I guess, to the public. We do all want answers as fast as possible, and things need to be designed very well because it is a very complex and heterogeneous disease, meaning that it's very different from person to person. And this is just to show you an example of the most common way in which we measure dysfunction or functional decline in ALS and how disparate that is from person to person. And so getting an answer can be very, very difficult. And this comes into eligibility criteria. Now, quite often I get asked, why is it that I'm not eligible for a given trial or any trial whatsoever? And again, it comes back to the fact that a clinical trial is a very expensive scientific study run by people who are investing money in getting an answer so that they can have return on investment. And so the design of a trial is not meant to exclude people out of a lack of compassion, but it is ultimately to maximize the chance of getting an interpretable answer, an answer that makes sense. And because of the heterogeneity, it is very hard to run trials well, but we have come a very long way. So just to give you an example of this, and I've just highlighted in bold there an example, let's say you have a small study and there are a lot of slow progressing individuals who get randomized into a placebo arm. You may actually not be able to see something that's working because those slow progressors are overwhelming the data in the treated group and looking like there's no actual effect, which is why we need to make sure we have good eligibility criteria, but also big enough and long enough trials that can actually get answers that are definitive. And again, they need to be long enough because you need to be able to see a difference between the treatment and the group who are not on the treatment. And it also has to be a point where there's change in the progression of an individual, but not so aggressive change. So someone who's a very fast progressor, that they're not going to be able to finish the trial, because if they can't finish the trial, it turns out that that's a data point that can't be used in the statistics. And ultimately it can cause a very big problem in determining in the end whether it works or not. Another question I get is why don't a lot of trials allow for a Darvone use while they will allow for Riluzole? And a lot of that just simply has to do with the fact that the use of a Darvone is fairly new in a lot of places, and in order for someone to incorporate that, they would have to go back and redesign and restart their trials. We could potentially see more trials in the future which will have a Darvone arms, but for right now, there certainly isn't a whole lot of them. One that I'll talk about next week is a Canadian trial called Pimozide that actually went back and did a delay purely so that they could restructure and allow people who are on a Darvone to be involved in the trial. Another very key question I get is why is there still placebo control? Many people feel it's unethical in ALS to have a placebo control. And as I said, these are answers that I don't necessarily want to give, but I'm trying to give you these answers because they're really the truth about where things are at. And one of the questions is, can we use historical controls? What about databases of placebo participants from other trials? And we do have databases like this. The question is, what historical controls do we use? Who chooses those? Do you let a company that stands to make a market and make funds off of a particular therapeutic to choose the right historical controls? And ultimately, by choosing those controls, you've just unblinded a group to be able to, that could lead to some sort of bias as to whether something works or Secondly, what about a specific one that has treatment regimen? For example, Brainstorm has a bone marrow extraction and then a reintroduction of stem cells into the spinal cord. How does that individually affect someone, and how do you decipher whether or not the actual therapeutic is working versus whether or not there's some a detrimental effect from the actual procedures happening? Which I'm not saying there is with Brainstorm, I'm saying is an example that we need to also balance out for the treatment regimen. And companies will be reluctant to use placebo controls in a lot of cases, or without placebo controls, because again, they're putting a risk out there with investments, and they need real answers to be able to prove ultimately that something is safe and it works. But there is a lot of work being done to reduce the placebo number as best as possible. We need to be able to do these in rigorous scientific trials, but we can do better in terms of the ratio of people on treatment to placebo. And I'll talk about a really cool future piece, which is coming up in a couple slides. But very quickly, I'm going to go through the way in which we tell whether something works is we try to find outcome measures that allow us to measure the progression of the disease and to be able to see whether or not there is a slower progression of a disease on the treatment arm versus the placebo, in certainly a blinded fashion. So one of the key pieces is it needs to be blinded to both the person doing the measurement, so the clinician, and also the person who is on the therapy or the placebo, because then we don't introduce any bias for anyone believing that something may be working or wanting it to work, because ultimately everybody wants every therapy to work. It's just, that we want them to work and get to people, but if that bias is introduced, it actually can lead to false positives or false potential to think something is working. So this is just a scale. You've probably heard of this before. It's got a score of 48, and as dysfunction occurs in different aspects of ALS, the score will reduce. However, there are certain nuances to this that can be problematic, and we are trying to improve on this. A good example would be somebody who may have speech and swallowing problems, and they may really reduce their score on those particular aspects. But what if throughout the duration of the trial they have no problems with their movements or their ability to walk or climb stairs, for example? They may actually decline and then plateau when that's not actually a real indication of what's happening at the progression of their disease. Another one called slow vital capacity is one that's being used as an outcome measure for certain muscle treatments that I'll talk about in a week. But there are a lot of alternatives I don't have time to talk about. We are trying to find more real-time ways to be able to study ALS progression in a day-to-day measure as opposed to different clinic visits. The last thing I'm going to talk about very quickly here, and I know I'm running a little over time, I apologize for that for anyone who has to go. Hopefully this will be something you can watch streaming. Is the need for biomarkers in trials. And I wanted to quickly take the opportunity to explain to you why those are so important. Actually, one of the big turning points in the field came from something I mentioned earlier, where a company called Biogen IDEC ran a big phase 3 clinical trial for ALS, 900 participants, which is huge, over 900. And in 2012, they had a pretty good excitement in the field that this might be something that could work for ALS. We'd seen in a phase 2 clinical trial, which was again underpowered, that there was a 20 to 30% slowing in disease progression, but there was tangible, cautious optimism in the field that this might be something that worked, and it ultimately didn't work in the phase 3 clinical trial. I apologize for that, a little mistake there on the animation. But it didn't work. But the problem was we also didn't have what's called a biomarker to make sure that this particular drug, dexpramipexole, was actually hitting its biological target. So we don't know if it didn't work because the drug didn't hit the right target, or we didn't know if the target itself is just not good as an ALS target for therapy. So this changed things a lot, and it meant that we really needed to move things forward. This is just again to show you that in phase 2, we really felt some cautious optimism, and it just didn't pan out. But very quickly about biomarkers in the last couple minutes here. One I'm not going to talk about in much detail here. Diagnostic biomarkers is one you'll hear about. This is not related necessarily to clinical trials, other than getting people on trials faster. These are ways in which we could hopefully find biomarkers to diagnose earlier. I'll give you a very fake example here, which can give you an idea of what these are. let's say someone has an initial hand weakness and you could get a blood test that can give you a definitive diagnosis and allow you to get on therapeutics or clinical trials at an earlier time point. But in terms of clinical trials, there are two really prominent biomarkers to talk about. One is a prognostic biomarker. This is a way in which we can help to understand progression of the disease better and get a biological readout to do so. So I'm going to give you a fake example. It's one that we are kind of working on, but it's still not something proven. So let's say neurofilament levels, which are a part of neurons, in the cerebrospinal fluid that bathes the brain and the spinal cord, are increased as the disease progresses, and we can measure that increase to see how the disease is progressing, because as the neurons die, they can spit out these neurofilaments. And if a treatment works, can we then tell that it's working by actually non-invasively taking that cerebrospinal fluid out and seeing that there's a reduction in those increasing levels of neurofilaments, or a reduction in the actual rate at which neurofilament increase is happening? It's something being worked on, but isn't definitively proven yet. But again, that's a way that we could use a prognostic biomarker as a readout for ALS. Predictive biomarkers are also very important because they may help tell us whether or not a drug is hitting its biological target, and also be able to help us understand who might be good for what particular therapies, and something called personalized medicine. And that's what we'll ultimately strive for, is that perhaps we can find specific cocktails of therapeutics in the future that will work for certain people. And if we can measure these biomarkers, we may be able to say, you are going to be good for this particular trial or this particular therapeutic. A couple of quick examples. I know I've hit 1 o'clock. These are the last couple slides. That trial I talked about before, Neuraltis, used biomarkers as an interesting way to a predictive biomarker for a trial. I showed earlier that they had a phase 2 clinical trial that showed about 27% of the people, when they looked at it afterwards, had what appeared to be an arrest of the disease for 6 months. When they looked at those individuals more closely, they said they had a high level of something called C-reactive protein, which made sense with what the drug was supposed to do. They then ran a phase 2 clinical trial where only the people eligible would have high C-reactive protein. And ultimately, while it didn't work, which is a good, again, warning for overinterpretation of phase 2 clinical trial results, it also was a good example of how we might be able to, in the future, try to understand what people might be benefiting from certain therapies. A really great example of this is one I'll talk about in a little more detail next week, which is an antisense oligonucleotide therapy of SOD1. These are people who have a familial or hereditary mutation in SOD1. And again, that's a great predictive biomarker for who might be good for this trial. By those who have the associated genetic mutation, those are the ones that will be eligible to be involved in the trials. And there's also a really good readout by having SOD1 protein levels in cerebrospinal fluid. So what they're able to see is that if you can then give this antisense oligonucleotide, which is meant to decrease the levels of SOD1, if in the CSF you can take this from a person who's on the trial and see a reduction in the SOD1 levels, what this tells us is if it works, we know that it's possibly or probably through reduction of SOD1 levels. But if it does decrease the SOD1 levels and it doesn't work for the actual therapy slowing disease, and that might tell us something about whether or not SOD1 is a good target by knocking this down. These are ways in which we need to better understand how an actual therapeutic is working in the body versus just seeing whether it works or not. An example of that would be a generic anti-inflammatory drug for ALS. 5 years ago, we would have just looked at whether or not it slowed functional decline or not. But in 2019, we really now need to have biomarkers that say, did it work for ALS? And then can we tell whether or not it worked or didn't work based on whether it's actually doing what it's supposed to do in the human body? So in this case, did it actually reduce inflammation? Can we find biomarkers that tell us that it was actually doing what it was supposed to do? And a lot of work has been put into this. And a few years ago, there was a big meeting in the United States, which had all of the top clinicians in ALS coming together to develop a set of guidelines on how clinical trials should be run in ALS. And those are available online now. And if you want to learn more about some of those, you can certainly look at that document. The last slide I have is how we really are working to advance clinical trials. Specifically, the Northeast ALS Consortium, NEALS in the United States, which have members in Canada as well. And largely led by Dr. Merit Sokovich at the Healy Center in Mass General, are trying to put together the very first what's called a platform trial for ALS. And the idea here is you can reduce the amount of placebos that are involved, and you can compare multiple treatments together into one clinical trial. So as long as companies decide that they're willing to do this alongside each other, you could have a particular trial where you've got a placebo arm, but then you've got 6 or 7 different therapies that you might be blindly randomized on. And then not only will that be better for people who want to be involved in experimental therapeutics to have reduction in the placebo arm, but it also is really important because it can speed up the process in which a company can find out whether or not they've got something they want to move to phase 3 or not. Let's speed up the whole process leading up to those definitive phase 3 clinical trials. And it's also designed in a way that if something fails out, you can actually plug another one in on an already existing clinical trial. That's just to say that there's a lot more information that can come from this, a lot more clearer way it can be explained by Dr. Sokovich. So stay tuned to more information and webinars on those platform trials. But that we are doing everything we can in the field to try to make it as efficient and effective as possible to get those therapeutics through to proof and into the hands of people living with ALS. So that's all I have for today. Next week, we're going to talk about specific experimental therapeutics. I know that's a lot. Hopefully, we can have this on demand for you so you can go back and look through some of those. And as I've said, the key thing is, what did I miss? What other questions do you have? Because this is the opportunity for us to sit and be able to have a discussion on questions people have about the various aspects of clinical trials. So thank you very much to anyone who's still out there and who's listening, and I welcome your questions as always. Thanks. Okay, so I have just learned that there are actually no questions. So, okay. Must have been very clear, but you know what, in all honesty, for anyone who's still out there, I just want to say again that the whole point is that we want to be able to make sure that that from everyone's standpoint, we understand that there are no easy answers, and really wish that we could find better ways to make all of this system happen, and we will continue to strive towards that. But also want to make sure that you are getting the information that you need so you can make the decisions that you want to make moving forward, and certainly alongside your clinician who feels the exact same way as I'm sure I've been saying. So please welcome your questions over the next week, and hope to see you next week for the second part of this webinar. Hope everyone has a lovely day, and I'll see you next week. Bye.